Hi, my name is Imelda Vertani, and I'm presenting with Rob and Tom. Our presentation is about Sanger's DNA sequencing method. This method has been discovered by Dr. Frederick Sanger in 1977 and has been one of the most widely used DNA sequencing methods. This method is done by the use of special bases called chain terminators or Dideoxy nucleotides. The difference between a Dideoxy nucleotide and a regular nucleotide is that the Dideoxy nucleotide doesn't have the hydroxyl group attached to the 3' carbon of its sugar. In regular nucleotides, this hydroxyl group allows the DNA polymerase to form a bond with the next nucleotide. And since the Dideoxy nucleotides don't have this hydroxyl group, they can't form any more bonds with the next nucleotide, so they stop the DNA synthesis. For a DNA sequencing to happen, first the double-stranded DNA needs to get denatured. Hitting the DNA molecule breaks the hydrogen bonds between the strands and separates them. The single-stranded DNA then gets used as a template for the sequencing. For Sanger's method, 4 reactions need to get set up. First, in each tube, we add a single-stranded DNA template, then a primer, a DNA polymerase, nucleotides, and also a small amount of Dideoxy nucleotides, which have been radioactively labeled by either 32 phosphorus or 35 sulfur. In each one of these tubes, the following happens. First, the primer binds to the 3' end of the single-stranded DNA template. Then, the DNA polymerase gets attached, and starting from the primer, it starts synthesizing new DNA by adding nucleotides to the chain. The chain continues until The chain continues until a Dideoxy nucleotide gets added instead of a regular nucleotide. And as we said, since the Dideoxy nucleotides are missing the hydroxyl group, they can't form any further bonds, so they stop the chain. And this process continues until all the nucleotides that have been added to the mixture get synthesized and there are no more free nucleotides. Because the Dideoxy nucleotides get attached to the chain randomly, the chains will have different lengths.
And so to measure the different lengths, the samples are ran through a gel electrophoresis, which we already learned, but each sample is placed in a well on the polyacrylamide gel, and an electric current is ran through the gel so that the fragments are separated by size. Smaller fragments will move faster and towards the cathode, the positively charged cathode, while larger fragments stay closer to the negative electrode. So here is an example of the polyacrylamide gel. You can see the smallest fragments down here near the bottom, near the positively charged cathode, and the larger fragments that don't move as far stay near the negatively charged anode. To read this, you would go in order from the bottom to the top, so whichever order it's in, and just go one by one to get the sequence. But although this picture is ideal and easy to read, the bands on an actual gel in the lab sometimes turn out to be blurry or with a lot of smudges, which make it hard to read. So they've been using capillary gel electrophoresis. So this technology has advanced, and it's basically a machine which will read the fluorescent chain termination markers that were placed on each of the different sampled tubes of the dideoxy ribonucleotide. The sample is set into the machine with positive and negative ends, and as the sample flows through a teeny tiny capillary gel tube, it is hit by a laser, which is read through a detector, and the results will be displayed on a computer in the form of a chromatogram. Each detection will have the primer, which is labeled in gray right here, and since they go in order by size, it will tell you the sequence of the sample one by one. So 1, and then this is 2 nucleotides after the primer, and then 3, and then 4. So the colored peaks in this chromatogram identify the order of the sequence. And so you can see the different colors which are associated with the different nucleotides. So today, Sanger's method of chain termination sequencing has largely been replaced by next generation sequencing because it is much more cost efficient and less time consuming for those large sequences. But because next generation sequencing is more expensive and the machinery is not widely available, it makes Sanger's method to still be utilized, especially for smaller sequencing and to verify results of the next generation methods. We found an article in the Journal of Molecular Diagnostics led by Amir Badad and his research team, which used Sanger's method of chain termination sequencing. Acute myeloid leukemia is caused by CCAAT enhancer binding protein alpha mutations, also known as CEBPA. They used a robust version of Sanger's method and in their study found 19 single mutations and 3 double mutations that were not identified with next generation sequencing. Sanger sequencing found 22% of the single mutations and 4% of the double mutations, which is kind of a lot if it's to dealing with someone's life. Here we have the articles we used to research Sanger sequencing.